Hello everyone and welcome to our module on bone. Let's start by talking about different types of bones. The long bones are bones in your body that support weight and allow movement. These are the bones that are found in your legs and in your arms. Flat bones are bones that protect organs. For example, your skull contains a number of flat bones. Short bones are the bones of the wrists and the ankle, and there are irregular bones, which are bones found in the vertebrae. You may know that the vertebrae have a very unusual and irregular shape. And then finally, sesamoid bones are bones that are embedded in tendons, and the classic example of a sesamoid bone is the patella or the kneecap. Now let's talk about the macroscopic structure of bones. The periosteum is the outer membrane that covers the outer surface of bones. It contains blood vessels, it contains sensory nerves. If you look at this picture on the screen here, the periosteum is this outer layer here seen surrounding the long bone, and it contains the blood vessels. Cortical bone is the outer surface of the bone. It's also called compact bone. Anytime you hear the word cortex or cortical, you should just think of shell. Basically, the outer shell of anything is the cortex. For example, the cortex of the kidney is the outside of the kidney, and in the same manner, the cortex of the bone is this hard outer layer of bone that surrounds the entire bone. It's the hard exterior bone. In addition to the cortical bone, there is another type of bone called trabecular bone. It's also called cancellous or spongy bone. This is the bone tissue shown in this part of the bone in this drawing on the screen here. This bone is soft and flexible, and it's found at the ends of long bones. It's trabeculated, which means it's sort of honeycombed with lots of spaces made by the bones. And what's important to understand about trabecular bone is that it has lots of surface area. Because the bone is curved, like drawn in this picture here, there's lots of surface area, and this will be important later when we talk about cells found on the surface of bone. And then finally, the medullary cavity, or the medulla of the bone, is the part of the bone that contains the marrow. It's usually found in the shaft of long bones. And you can see some of these basic bone structures on an x-ray, and they like to show you x-rays on the step 1 exam, so you ought to be able to find the basic structures of a long bone. For example, shown on the screen is a picture of the femur bone in the leg, which is a classic long bone. Note that it has this black section down the middle of the straight portion. That is the medullary cavity that contains the bone marrow. On either side of the medullary cavity, there are these thick white portions. That's the cortical bone. And then if you look up towards the top near the joint, you see that that black part down the middle has gone away, and instead you have these straight wispy lines. That is the trabeculated bone, and as I said before, it's usually found at the end of long bones, such as up towards the joint. There are some important terms related to long bones you should be familiar with. The epiphysis of a long bone is the end of the long bone. It's usually covered by cartilage. If you look at this drawing on the screen here, the epiphysis is here at the bottom and up here at the top. The metaphysis is the widening just before the epiphysis, so that's this portion right in here. And then finally, the diaphysis is used to describe the straight portion of a long bone, or the shaft that contains the medullary cavity. Let's talk about the types of cells you can find in your bones. Osteoblasts are the cells that synthesize bone matrix. We'll talk more about these cells in a minute. Osteoclasts are specialized macrophages. They're derived from circulating monocytes, and they secrete acid and protease enzymes that dissolve bone matrix. So it's an important principle to understand that your bone is constantly undergoing remodeling. There are osteoclasts that break it down, and there are osteoblasts that remake it all the time. It is not a static structure. The main two cell types to know about are the osteoblast and osteoclast, but there is a third type of cell found in bones. It's called an osteocyte. These are osteoblasts that have become buried in the bone matrix. They become osteocytes, and they play a role in controlling local calcium and phosphate levels. But the main two cell types that control bone turnover and bone remodeling are the osteoblasts and osteoclasts. So one of the things you can find in bone tissue are the cells that we talked about in the last slide, but lots of your bone is made up not of cells, but of something called bone matrix. This is the extracellular component of bone. It's synthesized by osteoblasts, the bone forming cells we talked about in the last slide. And lots of your bone matrix consists of type 1 collagen. In the cell biology section in the video on connective tissue, I talk in detail about collagen, and it's type 1 collagen that you find in bone. The other major component of your bone matrix is hydroxyapatite. This is a crystalline structure that contains calcium and phosphorus, and 99% of your body's calcium and 85% of the body's phosphorus is found in bone, and it's found in hydroxyapatite, which is made by the osteoblasts. When osteoblasts want to lay down new bone matrix, they first make something called osteoid. This is non-mineralized bone matrix. It's mostly proteins. It's laid down by osteoblasts, and later it becomes mineralized, which means calcium and phosphate are added to the osteoid. And this will be very important when we talk about bone disorders, because there are bone tumors and other bone disorders where osteoid is made in excess, and you need to understand that this is a proteinaceous material that is made by osteoblasts but has not been mineralized yet. Now we're going to talk about something that is incredibly high yield, and that is bone turnover. As I mentioned before, bone tissue is not static, it's constantly changing. Osteoclasts break down old bone, and osteoblasts lay down new bone, and there is a balance between formation and breakdown, and if this balance is normal, you will have normal bone structure and strength. But if there's too much breakdown, the bones will become thin, and if there's too much bone formation, the bones can become abnormally thick. What you need to understand is that this entire system of bone turnover is modulated by signals from the osteoblast. The osteoblast is really the controller of bone turnover, and the osteoclast is really just a slave to the osteoblast. These signals, which come from the osteoblast, some of them stimulate osteoclasts, some limit osteoclasts, and the combination determines the rate of bone formation and breakdown. The first signal from the osteoblast to the osteoclast that we're going to talk about is rank and rank-L. So rank stands for receptor activating nuclear factor kappa beta. This is a big, long, confusing name, but basically rank is just a receptor, and it's expressed on the surface of osteoclasts, as I've shown in my picture at the bottom of the screen here. When this receptor binds to something called a ligand, this leads to synthesis of nuclear factor kappa beta in the osteoclast, and that stimulates the osteoclast. So what binds to this receptor called rank? Well, the rank ligand, or rank-L. This is receptor activating nuclear factor kappa beta ligand. A ligand is something that binds to a receptor, and this one binds to rank, and it's expressed on the osteoblasts as shown in my picture here. So when osteoblasts make more rank-L, they can bind that rank-L to the rank on osteoclasts, and that is one way that osteoblasts can signal osteoclasts and create more bone breakdown activity by the osteoclasts. Two other ways by which osteoblasts control osteoclasts are shown in this slide. The first one is osteoprotegerin, or OPG. This is a decoy receptor for the rank ligand. It's made by osteoblasts, and it binds the rank ligand and prevents the rank ligand from binding to rank on osteoclasts. Osteoblasts can also make MCSF, which is macrophage colony stimulating factor. Osteoblasts can secrete this, and this will stimulate osteoclasts. So I've summarized everything with this picture on this slide, and basically if you know this picture, you can answer lots and lots of questions about bone pathology and bone physiology. So osteoblasts make rank-L, and they also make MCSF, and both of these things can stimulate osteoclasts. In addition, osteoblasts make OPG, which is a decoy receptor. It will bind to rank-L and block it from binding to rank. So when osteoblasts make more OPG, that decreases osteoclast activity. And I've summarized all this at the top of the slide. More rank-L, or MCSF, from osteoblasts leads to more osteoclast activity. More OPG from osteoblasts leads to less osteoclast activity. Now let's talk about how bone forms in utero. When a baby is born, that baby has long bones and flat bones and other bones. So how do those bones get there? There are two major ways by which this occurs in utero. The first one's called endochondral ossification, and the second one is membranous ossification. Endochondral ossification occurs during embryogenesis. This is the process by which our long bones form. They develop from hyaline cartilage that is secreted in utero from chondroblasts and chondrocytes. That's why it's called endochondral ossification. The way the bones are made is that a cartilage mold is first formed. These cartilage molds of our bones are called enlogen. For example, a long bone might start looking like a cartilage mold like this one on the screen here. As the mold grows, the chondrocytes die, and then osteoblasts come in from the blood, and they will turn this cartilage mold into a bone. The conversion of the cartilage mold into bone starts at two places. For example, if you have a bone that looks something like this, a typical long bone, at the center of the bone you will have what's known as the primary center of ossification. This is where osteoblasts will begin laying down bone matrix. At the ends of the bone, which is called the epiphysis, you will also get a center of ossification. This is called the secondary center of ossification. These two centers of bone formation will grow towards one another, and they will eventually meet and trap a layer of cartilage in a line like this. That layer of cartilage will become the epiphyseal or growth plate. And you may know in the bones of children, there is bone tissue with two plates at the ends, which are cartilage plates, and those are the remnants of the hyaline cartilage mold that was present in utero. And here's a picture from the web just to make this very clear in your mind. So we begin with a hyaline cartilage model, then the primary center of ossification opens up in the middle of the bone. Secondary ossification centers open up at the two ends of the bone, and those two ossification centers grow towards one another until we're left with a layer of cartilage that is trapped. That's called the epiphyseal plate or the growth plate. So now let's talk about the growth plates, which are also called the epiphyseal plates. These are found at the ends of long bones between the metaphysis and the epiphysis, which are those portions of long bones we talked about earlier. In fact, the word physis means growth plate. If you look at this X-ray image from the arm of a child in the bottom right side of the screen here, you can see the growth plate right here. These growth plates contain hyaline cartilage, and they have chondrocytes which grow towards the epiphysis. So the chondrocytes are going in this direction away from the center of the bone. The osteoblasts are laying down bone matrix towards the center of the bone, and as the child grows, the growth plate will move towards the joint, with bone being laid down behind the growth plate towards the center of the bone. Now at puberty, the growth plate closes and the epiphyseal lines will be formed, which will be present throughout adulthood. So if you look at this picture at the top of the screen, which is from the knee, you can see this line right here. That's the epiphyseal line. That's the remnant of what was the epiphyseal plate before puberty. We'll get back to the in utero formation of bone tissue in a minute, but let me pause and mention woven and lamellar bone. Woven bone is the first type of bone formed by osteoblasts. It's also called primary or immature bone, and it contains disorganized collagen fibers, and it's relatively weaker bone. Later, this woven bone is remodeled or replaced by osteoblasts, and it becomes lamellar bone. Lamellar bone is layered and organized and stronger. So at the growth plates in children, you will initially see osteoblasts lay down woven bone, and then later it will become lamellar bone. You can also see woven bone in adults if there's an injury. This is the type of bone that osteoblasts will initially lay down at the site of a bone fracture or injury. So back to our discussion of the in utero formation of bones. The second process by which bones are formed in utero is called membranous ossification. In this type of bone formation, bone matrix is formed directly and not from a cartilage mold. Osteoblasts will lay down woven bone, they'll later remodel it to lamellar bone, and this is the way most of the flat bones in our body are formed. For example, the calvaria, which is the skull bones, many of these are formed by membranous ossification, same thing for many of the facial bones. If you understand endochondral ossification and growth plates, then it's very easy to understand achondroplasia. This is the most common cause of dwarfism. It is a genetic disorder caused by a gene mutation in the fibroblast growth factor receptor 3 gene. 80% of cases are due to a spontaneous mutation, but this can run in families, and when it does, it is autosomal dominant. By that I mean that anyone with one copy of the gene will develop dwarfism. If you have two copies of the gene and you are homozygous, you will not survive. Babies who are homozygous are stillborn. So if you see a patient who has dwarfism from achondroplasia, that patient must be heterozygous and have a single abnormal copy of the gene. If they were homozygous, they would not be alive and seeing you in clinic. Basically, achondroplasia is a disorder of defective endochondral ossification. The mutation that occurs in the gene is a gain of function mutation, so the growth factor receptor is activated and turned on. Now you might think a turned on growth factor would lead to more growth, but in fact what happens is chondrocyte proliferation is inhibited. As a result, the long bones do not grow, so people with achondroplasia are dwarfs, which means they look like this man in the picture. He has very short arms and very short legs, but his torso and his face are normal, and that's because most of the bones in the torso and face do not develop by endochondral ossification, but the long bones of the arms and the legs do. This is a very simple disorder to understand if you just understand endochondral ossification. In the biochemistry section in the video on the lysosomal storage diseases, I talk about a group of disorders called mucopolysaccharidoses. These are disorders like Hunter syndrome and Hurler syndrome, where children have an enzyme deficiency and they cannot metabolize mucopolysaccharides like heparan and dermatan sulfate. The reason I'm mentioning this here is because chondrocytes are cells that degrade lots of mucopolysaccharides. So in children born with one of these disorders, the mucopolysaccharides accumulate in the chondrocytes and the chondrocytes die. So as you can imagine, this leads to similar problems to achondroplasia, problems at the growth plate and with growth of long bones, and short stature and malformed bones are common in these disorders. In the basic science laboratory, when researchers are studying osteoblasts, there are a number of commonly used activity markers that are studied. These are substances whose levels rise when osteoblasts are more active. These are also sometimes used clinically to make the diagnosis of a disorder in which osteoblasts are overactive. The first activity marker is alkaline phosphatase, which I'll talk about in the next slide. The second one is osteocalcin. This is the major non-collagen protein found in bone matrix. Most of the bone matrix is collagen, but the major non-collagen protein is osteocalcin, so when its levels go up, that means osteoblasts are working harder. The third activity marker is type 1 procollagen. So in the cell biology section in the video on connective tissue, I talk about the synthesis of collagen, and I talk about how it's initially synthesized as 3 pro-alpha chains combined together. This is called procollagen. It looks like what I've shown on the screen here. This is what is secreted from osteoblasts, and then it is modified extracellularly into tropocollagen and collagen. So you can imagine that when the levels of type 1 procollagen go up, that means more pro-alpha chains are being secreted from osteoblasts, and therefore that is a good activity marker of osteoblasts. Alkaline phosphatase is an enzyme that's found in bone tissue and in the liver, although there are different forms, and levels of alkaline phosphatase in the blood can rise in bone disorders or liver disorders. There's also a placental form called appropriately placental alkaline phosphatase, and you can see high levels of this in the blood in some germ cell tumors. The reason this is important in bone is because it's one of the major proteins found in bone tissue. It's found bound to osteoblasts and also free. And what it does is it creates an alkaline environment around the osteoblast. And if you can remember that alkaline environments are favorable for calcium deposition, that's very helpful to understanding bone physiology. The osteoblast creates a high pH alkaline environment around the cell, and that makes a favorable environment for calcium to settle out and form crystals of hydroxyapatite in bone. And if you can remember that an alkaline environment is favorable for bone formation, that's an easy way to remember that acidosis is bad for bone formation. An acidotic environment is unfavorable for the deposition of calcium crystals. In addition, acidosis stimulates osteoclasts, and you can see hypercalcemia as a complication of chronic acidosis from bone breakdown. And patients who have a chronic acidosis can also develop reduced bone mineral density and osteoporosis. And this sometimes complicates patients who have renal tubular acidoses. These patients are chronically acidotic, and that can have negative effects on bone formation. I talk about parathyroid hormone in detail in the endocrine section in the video on the parathyroid gland, but I'll review its role in bone physiology here. So parathyroid hormone has multiple effects on bone. It can stimulate bone resorption and formation. The dominant effect varies depending on the dosage and timing. If you administer parathyroid hormone to bone tissue in a steady, continuous manner, this will lead to bone resorption and bone breakdown, and this will raise the serum calcium. And this is the way parathyroid hormone works physiologically to maintain calcium in our body. If, however, you artificially administer a low-dose once-daily bolus of parathyroid hormone, you will see the opposite effect. The bone mass will go up and there will be bone formation. The reason that parathyroid hormone does this is not well understood, but because of this different effect of a once-daily bolus, you can use artificial parathyroid hormone. It's a drug called teriparatide to treat osteoporosis. You might think that giving parathyroid hormone to someone with osteoporosis, which is a disorder of weak bones, would be bad, but it turns out to be good because the once-daily administration of a bolus works differently than the continuous steady infusion that the parathyroid gland does to the bones. So parathyroid hormone has different effects depending on whether you administer it continuously or as a single bolus. In addition, it affects the bone compartments differently. So cortical bone, which is the hard outer shell bone, decreases in response to continuous parathyroid hormone administration. So when your parathyroid gland releases continuous PTH, you see loss of cortical bone, and that's what raises the serum calcium level in response to parathyroid hormone from the parathyroid gland. Trabecular bone, on the other hand, increases in response to intermittent low-dose boluses of PTH. This is the trabecular portion of the bone here, and it's very useful that this effect of parathyroid hormone occurs on trabecular bone clinically because you can administer recombinant parathyroid hormone, that's teriparatide, and this will increase the bone density of trabecular spongy bone. And that's the type of bone that often decreases in osteoporosis. There's lots of it in the spine. There's also lots of it in the head of the femur. And these are places where people with osteoporosis have lots of fractures. So if you give recombinant parathyroid hormone, teriparatide, you will see a rise in the bone density of the trabecular bone, and it's very helpful for treating osteoporosis. Let's talk about how parathyroid hormone affects the different cell types in bone. Osteoblasts contain PTH receptors, and this is why low-dose intermittent boluses of PTH can increase bone mass. That's because the PTH can directly stimulate the osteoblasts. Osteoclasts have no PTH receptors. They are activated indirectly by osteoblasts, and this follows the principle we talked about before, that the osteoclasts are really slaves to the osteoblast. So the way that parathyroid hormone leads to breakdown of cortical bone and a rise in the serum calcium is by activating the osteoblasts, which in turn will activate the osteoclasts. And they do this through the mechanisms we talked about before. Osteoblasts can make MCSF and secrete that. They can also express RANK-L on their surface, and both of these will activate the osteoclasts, and that's how parathyroid hormone can lead to bone breakdown. I'll finish this video by talking about estrogens. Estrogens have numerous effects on bone. First of all, they close the growth plate at puberty. So in girls at puberty, the ovaries begin making lots of estrogens, and that will close the growth plate. In boys at puberty, the testes make lots of androgens, but they also make estrogens, and those estrogens will also close the growth plate in boys, just like in girls. A second important effect on bone of estrogens is that they raise bone density, and this is why women often develop osteoporosis at menopause, because their ovaries stop making estrogen, estrogen levels fall, and that leads to a fall in bone density, and that leads to osteoporosis. The way that estrogens raise bone density is by decreasing the activity of osteoclasts. Remember, there's always a balance between bone formation and bone breakdown. If you decrease the amount of bone breakdown by osteoclasts, you will get relatively more bone production, and that's what leads to increased bone density. Estrogens have been shown to decrease osteoclast activity and even induce apoptosis of osteoclasts. And the way they do this is through those same signaling systems we talked about earlier. Estrogens have been shown to stimulate OPG synthesis by osteoblasts. Remember, OPG is the decoy receptor for rank L, so more OPG leads to less osteoclast activity. In addition, estrogens have been shown to decrease MCSF release by osteoblasts. They've also been shown to decrease rank production by the osteoclast so that the receptor for rank L is not present. And through all these mechanisms, you get less activity of osteoclasts, and that's why estrogens are good for bone formation. And that concludes our video on bone.